Hello students, this is Mrs. Shweta Laddha from your biology teacher. We had started with chapter 1, Reproduction in Lower and Higher Plants. In this chapter we had started with pollination, types of pollination. If you remember, what is pollination? Pollination is transfer of pollen grains from anther to stigma. Then pollination is of different types, self-pollination, cross-pollination. Self-pollination we studied about autogamy and Dichogamy. Autogamy is transfer of pollen grains from the anther to the stigma of one flower, same flower. Dichogamy is two flowers on the same plant. In cross-pollination allogamy we studied xenogamy, when transfer of pollen grain is taking place between two plants but they should be of the same species. And in hybridization we studied it can be of different species, different varieties. After that, we started with agencies of pollination, abiotic, biotic. In abiotic, there is wind pollination, Entomophily or Hydrophily. And in biotic pollination, there are many other agencies. We'll study about them in detail. Last time, if you remember, I had already finished teaching you Entomophily and Ornithophily. Today, we'll proceed and learn the remaining types of pollination. So, let us start with our PowerPoint presentation. So today we'll start first with entomophily. The word entomophily, it is derived from entomon, means insect. So the pollination here takes place by insect and it is called as entomophily. In this case, the plants are called as entomophilous plants. Moths, butterflies, most usually honeybees, beetles, flies, they carry out entomophily. Flowers, they should be large, colored, beautifully colored to attract the insect. They have fragrance, beautiful, pleasant fragrance and lots of nectar so that the insect can come to have food over them. Sometimes modifications are made. Corona in passion flower, petaloid bracts in bougainvillea. Bougainvillea, the colors you can see in bougainvillea, actually flower is white in color. The bracts become colorful to attract the insects for pollination. In passion flower you might have heard about this flower. We commonly called it as Kaurav Pandav fool. There are so many petals. Beautifully designed corona is there just to attract the insects. Pollen grains have rough or spiny exine. Always remember students, in wind pollination, the exine is smooth. In flowers which are carrying out they are pollinated by insects, they have a rough or a spiny exine. The stigma are sticky, okay, so that the pollen grains which are attached legs of the insect, they get stick to the stigma. Example, entomophily occurs in jasmine, in rose, salvia, bougainvillea, etc. So this is entomophily, having large colored, fragrant, pleasant fragrance flowers, having lot of nectar, and pollen grains have rough or spiny exine. Stigma is sticky. This is a wonderful example of liver mechanism which is seen in salvia. Flower is bilipped. Bilipped, there is this upper one lip and then there is a lower lip. So bilip flower is present in salvia, and let me show it to you. So this is upper lip and this is the lower lip. Bisexual flowers. Bisexual, it is having both the androecium and gynoecium. But the flower is protandrous. Protandrous, androecium matures first, that condition is called as protandry. Gynoecium matures first, that is called as protogamy. But here it is protandrous, androecium is maturing first. And bifurcated or bivaricate anther. This is interesting. Now anther, basically it consists of filament, connective and anther, isn't it? In this case, the filament is small and the connective is bifurcated to form two lobes. Upper lobe, which is a fertile lobe, and lower lobe of the anther is sterile lobe. So such type of anther, it is bivaricate or divaricate or bifurcated. Who is bifurcated? The connective is bifurcated to form upper lobe, lower lobe. How is the upper lobe? It is fertile. How is the lower lobe? It is sterile. So let us see what happens in this flower. Stamen shows liver mechanism. Liver mechanism. Suppose the insect is visiting this flower. When it visits its flower, accidentally it pushes this lobe down. When it pushes this sterile lobe down, the upper fertile lobe, it presses on its back. You can see in this picture. So this insect is visiting the upper fertile lobe, is dusting the pollen grains on the back of the insect, isn't it? And by this time, as it is protandrous condition, anthers are mature, pollen grains are formed, but gynoecium is not mature. So self-pollination will not take place. Now this flower, it goes, this honeybee, it goes and visits another flower. When it is visiting another flower, that time the gynoecium is mature in that flower. And so what, and the stigma is sticky. So they will stick, they will collect the pollen grains which are deposited on the wall of the honeybee, and thus cross-pollination occurs with lower sterile anther lobe and upper fertile anther lobe. I hope you understood this concept. So this is like a liver, this is a lower lobe, this is a upper lobe. Upper lobe is fertile of the androecium, lower lobe is sterile. Insect is visiting. When it is visiting, it is pushing this lower sterile lobe. The fertile lobe bends down and it dusts all the pollen grains on the back of the insect. Now this insect goes and visits another flower. In another flower, the gynoecium is matured. The stigma is sticky. These dusted pollen grains on the back of the insect will stick to the stigma, and thus cross-pollination takes place. When insect visits the flower for nectar, it presses the sterile lobe. Upper fertile lobe dusts pollen grains on the back of the insect. Now pollen grain covered insect pollinates the stigma of another flower whose anthers are withered, because by that time when the gynoecium are maturing, the androecium, the anthers withers, because protandry condition. First androecium is, what we'll say, is getting fertile, or they are getting matured, and then the gynoecium gets matured. So a wonderful liver mechanism can be seen in Salvia. Who acts as a liver? The anther acts as a liver. How is the anther? It is divaricate. Who is divaricate? Who is branch? The connective is branch. Lower sterile, upper fertile lobe. This is a very interesting, can be asked for 2 marks in your exam. Salvia liver mechanism. I hope you followed. Now we will discuss about the next type of pollination, which is called as Chiropterophily or Chiropterophily. So this pollination, it takes place by bats. So now we know bats are nocturnal. They are active during the night time. So flowers also, they are very large, they are very stout, so that they can hold the weight of the large, huge bats, and they are nocturnal. These flowers, they bloom, they open during the night time. And the flowers, they're emitting very rotten fruit-like, strong, fermenting, fruity odor, because the bats, they like these bad, emitting rotten fruit-like, very strong odor. So the flowers are emitting such type of odor. Flowers are dull colored, you can see in the picture, and they produce copious means abundant, bharpoor nectar, because the bat, they drink lot of nectar, they are hungry, and they will just visit one, they will drink all the nectar, and they should be fulfilled, they should be satisfied. So flowers are producing copious, abundant nectar. Large number of pollen grains are produced because chances of pollination or chances of wastage are more. Pollination is less. Example, it occurs in Anthocephalus, which is called as Kadamba flower, Kygelia pinnata, sausage tree, Bahunia, Adansonia, or the baobab tree. So this is Chiropterophily, that is pollination when it is occurring by bat, and you can see how the flowers are adapted according to the bat, habits of the bat, that is they are nocturnal and producing fruit-like, strong, fermenting, fruity odor and abundant nectar. So I hope this is also clear to you. Now we will discuss about hydrophily. Hydro, water. So when the pollination it is taking place by water, it is called as hydrophily. Water pollinated flowers are called as hydrophilous plants. Flowers, they are unisexual, staminate, pistillate flowers, unisexual flowers. They are very small. Water does not need to be attracted. So no need to have big flowers, attractive flowers, no colors. So small flowers, inconspicuous flower, not so beautiful flowers. Pollen grains and other floral parts are coated with mucilage. This is very, very important character. They should be coated with mucilage so that they remain unwettable. They don't get rotten when they are in water. So they are aquatic plants, aquatic flowers, so they should not get rotten, and therefore they are covered by mucilage. Specific gravity of pollen grain are controlled by the starch in the pollen grains. Most of the time asked in MCQ, who is controlling the specific gravity of the pollen grain? There is starch in the pollen grain which controls the specific gravity, and because of which the pollen grains, they can swim. Stigma is very long and it is sticky. We'll see how helps when the stigma is long and sticky when it is pollination taking place by water. Hydrophily is of two types. Epihydrophily, also called as Epihydrogamous. This work is in NCERT books. Even if you remember epihydrophily, that is enough. Means pollination is taking place on the surface of water. And hypohydrophily, hypo means below, pollination when takes place under the water. So two types of hydrophily we will be discussing. Epihydrophily, above the water. Hypohydrophily, under the water. First, let us discuss about epihydrophily. You can see this water pollination in Vallisneria. Pollination takes place on the surface of water. This is important. Where is the pollination taking place? On the surface of water. Therefore, it is called as epihydrophily. Example, Vallisneria. It is a dioecious plant. Dioecious. Male and female plants are separate. You can see in the picture. This plant A, suppose this plant B. Here what do you see? Staminate flower. So it is a male flower. What do you see here? Pistillate flower. So it is a female flower. So it is a dioecious. When the staminate and pistillate flowers are borne by different plants, such plants are called as dioecious plants. Therefore, I had given the definitions earlier. Example, Vallisneria. The male flowers detach. You can see in the picture how these staminate. The male flowers are detaching. They are separating. And they start floating on the water. You can see they start floating on the water and they release sticky pollen grains. They are releasing pollen. These are the male flowers which are floating on the water surface. They start releasing sticky pollen grains. The female flowers have long stalk and sticky stigma. Look at this stalk, how it is long. So flowers, female flowers, they are having a very long stalk and they have a sticky stigma. Obviously, when it will uncoil itself, reach the surface, it will attract the staminate flower and thus the pollination will take place on the surface of water. Next, we will discuss about hypohydrophily. Hypo means under, hydrophily, pollination when it is taking place under the water. Now, pollination takes place below the surface of water, unlike Vallisneria, where it was a dioecious plant and pollination occurred on the surface. Here, pollination takes place below the surface. It takes place in case of Zostera and Ceratophyllum. Remembering example is very important. They may ask you for match the pair, give the example, MCQs. So you should remember, learn the examples by heart. So in Zostera, Ceratophyllum, hypohydrophily takes place. In Vallisneria, epihydrophily takes place. It is a monoecious plant. See the difference also you can write. In epihydrophily, it is a dioecious plants. Plants, male plants, pistillate, staminate plants are separate. In monoecious plant, both the staminate and the pistillate flowers are produced on the same plant. Look here, there is only one plant A. Here the pistillate flowers are shown. Here the staminate flowers are shown. So male and female flowers are produced separately but on the same plant. Therefore, it is a monoecious plant. And here we are studying the example of a freshwater hydrophyte. The staminate flower produce 30 to 40 stamens. So 30 to 40 stamens are produced. The male flowers, they float to the surface and dehisce. So they will float to the surface and they will dehisce. You can see in this picture I have taken from NCERT books. So this is the male flower which is dehiscing, which is opening. Dehisce means what? They open to release the pollen grain. Stigmas, they are long and sticky. Look at this picture. You can see this is the stigma which is long and sticky. Okay. They will catch the pollen grain by swaying action. They sway in the water like this. They will sway and they will catch the pollen grain and they will bring it down under the water and pollination will take place, fertilization will take place under the water. So it is hypohydrophily. So I have completed here epihydrophily as well as hypohydrophily. Now let us see what are the advantages of self-pollination. If self-pollination takes place, what are the advantages? Self-pollination, it is a sure method. Chances of failure is very less because the pollination is taking place within the flower or two flowers but on the same plant. So it is a sure process of pollination. It is economic. Flowers do not have to modify themselves. They don't have to produce large flowers, attractive flowers, fragrance, nectar. No, like ornamental things they don't have to produce. So there is less wastage of energy. It is economic. There is least wastage of pollen grains. They do not have to produce large amount of pollen grains. Genetic stability can be obtained by preserving the desired characters. And these are the advantages. Let us see what are the advantages of cross-pollination. Cross-pollination, they includes genetic variation, because here two different species are going to mix with each other. So genetic variation is there. Recombination occurs. So it causes variation. It brings about variation. Undesirable characters. The characters which are not needed, they can be eliminated by hybridization. Improved varieties can be produced by hybridization. Better variety having more vigor, more vitality, disease resistant variety, variety which are better adapted to the climatic conditions. Such type of offsprings can be formed. It favors variation, and as there is mixing of genes, mixing of characters, it favors to variation. Variations are brought, new characters are developed, and thus it favors evolution. So these are the advantages of cross pollination. Now suppose a question comes, what are the disadvantages? Of course you'll write exactly opposite. Whatever were the advantage of self pollination are the disadvantages of cross pollination, and whatever are the advantages of cross pollination are disadvantages for self pollination. So I hope you have understood pollination, agencies of pollination, types of pollination, and advantages and disadvantages of pollination, both self and cross pollination. Now, do you know that in all breeding programs, the plants are hand pollinated to ensure cross pollination between selected varieties? You have a practical also based on this topic, where you will be studying the 3 steps of hybridization. First, you are selecting a good variety. Then you are emasculating the flower. Emasculation is removal of anthers before anthesis. Removal or cutting of anthers from a flower before it produces pollen grain, so that the flower becomes a female flower, is emasculation. Then hybridization is carried out by hands with a brush. Then it is covered with a polythene bag, which is called as bagging. Tagging is done and hybridization is done. So breeding programs, the plants are hand pollinated so that cross pollination between selected varieties is brought. It can be carried out in wheat and rice. Now let us start with the next very important part of this topic, part 4. Here we will be discussing about outbreeding and inbreeding devices. Outbreeding device. These are adaptations. Outbreeding devices are also called as contrivances, or they are called as adaptations. Why? And who is developing? Flower is developing. For what? So that they can promote cross pollination. This is called as outbreeding device. Adaptations or contrivances developed by the flower to promote cross pollination. Means the flower is helping, only cross pollination should take place, self pollination should not take place. This is called as outbreeding, cross pollination. Changes, adaptations, contrivances in the flower so that they promote cross pollination. We will be studying 7 types of outbreeding devices. First is Dichogamy. Very important topic I am starting now. It is also called as unisexuality or Dichogamy. Here, flowers are unisexual. If flower is unisexual, you can see in the picture, this is a female flower, this is a male flower. Is there any chance of self-pollination? No, they have to undergo cross-pollination only, because one flower is a female flower, other flower is a male flower. I hope you remember what is a male flower called? Staminate flower. What is a female flower called? Pistillate flower. If the flowers are unisexual, staminate and pistillate, of course cross-pollination is promoted. It takes place in papaya, it takes place in mulberry. Dichogamy or unisexuality, first outbreeding device. Flowers are staminate or pistillate. Second is Dichogamy. Now what is this Dichogamy? Di means 2. Gamy is the maturation time. So in bisexual flowers, the time of maturation of anthers and stigma is different. There are 2 types. Protoandry, when androecium matures first. Just now we studied the example of Salvia having protoandry. Salvia, cotton, sunflower. Protoandry. In this flower, the flower is bisexual. They are having both androecium and gynoecium. But androecium anthers mature first. Gynoecium does not mature. And protogamy, when carpels mature earlier than the stamen. So, of course, if the time of maturation of androecium and gynoecium is different, it is going to promote cross-pollination, outbreeding. So, first we studied Dichogamy. Dichogamy is unisexuality. Flowers are pistillate flower or staminate flower. Then we are discussing Dichogamy. In Dichogamy, we are studying protandry condition, protogyny condition. Protandry, anthers are maturing first. Protogyny, carpels are maturing first, and therefore cross-pollination is promoted. Example of protandry, salvia, cotton, sunflower, disfloret. Examples of protogyny, magnolia, mirabilis jalapa, ranunculus, gloriosa. These are the example of protogyny. So next, the third is self-sterility or self-incompatibility. You can look at this picture. Pollen grains of a flower are incapable of affecting fertilization even if placed on the stigma of the same flower. You can see in the first picture, S1 and S2 both are. So, as the pollen grains are germinating, they might be from different type of flower. So, in this you can see this is not able to germinate. In this also they are not germinating. So, this is self-incompatibility. If the pollen grains from the same flower falls on the stigma of the same flower, the pollen will not germinate on genetically similar individuals. And when the pollen grains do not germinate, when the pollen grains of the same flower falls on the stigma of the same flower, this is called as self-incompatibility or self-sterility. So, in simple language, if you take anthers, this is one flower. This flower is having, say, anthers, and this is the stigma, style ovary. You take some pollen grains, you dust it on the stigma, they will not germinate, they will not form pollen tube. This is self-sterility or self-incompatibility. It occurs in potato, in tobacco, in petunia and thea. So self-sterility. First we started with Dichogamy. In Dichogamy we studied pistillate flower, staminate flower. Then we studied Dichogamy, protandry, protogyny. Third we studied self-sterility or self-incompatibility. Fourth we will study is prepotency. In many plants like grapes, pear, apples, pollen grains from one flower when present on the stigma of another flower will germinate more quickly as compared to the flower of the same species. Means if this is one flower, this is another flower. If the pollen grain is falling on the stigma of the same flower, germination will take place, but it will take more time. But here it will germinate quickly. So this is prepotency. So fourth is prepotency. After this, we will discuss about heterostyly. Very interesting example of heterostyly we'll study in oxalis, in primrose. Occurrence of two or more types of flowers, and they are having different length of the styles and stamens. Look at this picture. First flower, A flower, B flower. Look at the height of the anther in this. Look at the height of the anthers here. Look at the height of the carpel here and carpel here. Similarly, in this we are having three varieties of flower in oxalis, A, B, C. Look at the height of gynoecium here, here, here. Isn't it different? Look at the height of the anthers. First height, second height, and these are so tall. So different height are present. Heterostyly or heteroanthy. In jasmine and primrose, there are two types of flowers, but in oxalis, there are three types of flowers. So you can say this is the example of jasmine or this is the example of primrose, where there are two types of flowers. In oxalis, there are three types of flowers. Insect visiting a flower will get dusted with the pollen grains on a particular area of its body, depending upon the height of the anther, how deep it is going. It will depend how deep it is going, and then only it will be able to bring about the pollination. So here also it prefers cross pollination. It is also outbreeding device. Here look at this. This anther is the height of the anther and the height of the stigma is so far away. So pollination will not take place. Heterostyly, in case of heterostyly, insect when visiting a flower, it gets dusted depending upon which part of the flower, the height it is visiting and where the pollen grains are dusted. So fifth was heterostyly. Now this insect will pollinate the flower in which stigma lies at the same level of the anther, isn't it? If anthers and gynoecium at the same level and the insects are visiting, then only pollination takes place. So next is heterostyly. This one, the sixth one is Herkogamy. Very interesting. It can be seen in case of Calotropis. Herkogamy. Let us see what is Herkogamy. In some flowers, there may be some physical barriers present in the anther and the stigma, so that pollination between them becomes difficult or impossible. Look at this picture. You can see a labeling called as gynostagium, Actually, if you look at this picture properly, these are the anthers. This is the gynoecium. Don't you think the anthers and stigma, they are so close to each other? See, they are connected to the stigma and forming a gynostagium. You might be thinking that the anthers and gynoecium are so close to each other, easily self-pollination will take place. But no, self-pollination does not take place because the pollen grains are not present directly in the anther, but the pollen grains are present in such sac-like structures which are called as pollinia. And so there are barriers between the stigma and the anthers. The pollen grains are not present directly in the anther lobes, but they are present in sac-like structures which are called as pollinia. And so self-pollination is not possible. In Calotropis, pollens are present in sac-like structures which are called as pollinium, and pollination is completely dependent on the mercy of insects. You can look here. If the insect is visiting this flower, if it is sitting on the gynostagium, claws they get attached, they get, we'll say, hooked to these pollinium, and when they carry this pollinium, of course what this pollinium is consisting of, it consists of pollen grains. Herkogamy, physical barriers present between the stigma and the pollen grain. Simple words. Presence of physical barriers between anther and the stigma. Who is the physical barrier in Calotropis? The pollinium is present. Last one is suppression of one sex. In certain bisexual flowers, one sex organ, either stamen or carpel, is completely suppressed. It is sterile. Now suppose a flower is bisexual, androecium, gynoecium, both are present, but one is sterile. Of course, then what will happen? Cross-pollination will take place. So, suppression of one sex, sterility of one sex will always promote cross-pollination. Another does not form pollen grains, and or sometimes it should be anther. Anther does not form pollen grains, or carpel does not produce ovules. So one is suppressed, other is functioning. This will promote cross-pollination. Thus, it facilitates cross-pollination. Let us quickly see what are the adaptations for autogamy. When flower opens to expose its sex organ, it is called as chasmogamy. Now we are saying adaptations for self-pollination or autogamy. First we studied adaptations or contrivances for cross-pollination. Now we are saying adaptations ki self-pollination will take place. So when flower opens, exposes sex organ, it is called as chasmogamy. When anthers and stigma of a flower, they bloom, they mature at the same time, it is called as homogamy. Some flowers are self-pollinated even before the opening of the flower. Such flowers are called as cleistogamy. And you can see such flowers, underground flowers in Commelina bengalensis. Devices for favoring self-pollination, homogamy, cleistogamy. When carpels and anthers, they attain maturity at the same time, homogamy. Cleistogamy, when flower does not open, so of course self-pollination will take place. Example, geocarpy is seen in groundnut flowers, viola pansy, oxalis and commelina. In commelina, you can see both type of cleistogamous flower and chasmogamous flowers are seen. Cleistogamous flowers, they don't open, so self-pollination take place. Chasmogamous flower, they open but they mature at the same time, so self-pollination occurs. So we have seen about adaptations for self-pollination. So students, we have studied very important topics today. We have studied different types of pollinations, insect pollination, wind pollination, bird pollination, Chiropterophily, bat pollinations, Hydrophily, Epihydrophily, Hypohydrophily, and then we studied contrivances that are adaptations for outbreeding as well as inbreeding. Always remember, outbreeding 7 types are there, inbreeding only 3 types are there. Homogamous flowers, where you will discuss about chasmogamy and cleistogamy. Hope you have understood. Do take down the notes, revise this part. Next time we'll continue with this chapter. Thank you.